Remember from last time? What were the take-home messages from the last presentation? You were preparing for the exam, obviously. So. Come on. Yes?
Differences between gram-positive and gram-negative bacteria?
That was one thing. Exactly. The outer membrane, the cell wall of these two bacterial types. What is it that differentiates them? Say?
The thickness of the peptidoglycan.
Yep. Peptidoglycan layer, much thicker in the gram-positives. And it's exposed, while the peptidoglycan layer in the gram-negatives is embedded within the membrane in the periplasmic space. And when you learn about antibiotics, you learn in the gram-negative bacteria, the penicillin binding proteins are within that periplasmic space, so that the antibiotic has to penetrate through the outer membrane to get through the peptidoglycan in order to bind to the penicillin binding proteins. So this is another application. If you know the basic structure of the bacterial outer membrane, you can apply it in regards to antibiotics, for example. What else? See, I do this intentionally so that when the time comes for review, you will already have mastered this stuff. Yes?
You talked about like different PAMPs and toll-like receptors.
Yes. Okay, what do you remember from the PAMPs and the PRRs? Yeah, so PAM, pathogen-associated molecular patterns. What does that mean? Yes?
They're gram-positive bacteria produce a response to like sugar.
So gram-positive, but gram-negatives also have them. PAMPs, what are PAMPs? What are pathogen-associated molecular patterns? Yes?
Components of like the cell wall which will alert the immune system of the presence of
Exactly. So these are components, they can be components of anything in the bug. But the take-home, the most important thing is that those are components that are foreign to us. We don't produce them. So when we don't produce something and it's recognized by cells, our cells, then there's a reaction to them. Right? That's what you need to understand. There's a reaction to these things. So these are foreign agents, foreign molecules that are produced by these bacteria that come into play with our cells. And what are the PRRs? Pattern recognition receptors. Those are receptors. That means they're on our cells. And they recognize the PAMPs. And when they recognize the PAMPs, what happens? You get an activation of what kind of response? Innate. An innate immune response. That's the take-home message. PAMPs, PRRs, an immediate innate response. How is that response activated? What happens intracellularly within the cell? What did I talk about in terms of a transcription factor? NF-kappa B, nuclear factor kappa B. That is at the heart of inflammation. Immediate innate responses. Because the moment PAMPs are recognized by the PRRs, those PRRs activate a pathway within the cell that leads to the release from the cytoplasm of nuclear factor kappa B, NF-kappa B. And when NF-kappa B gets released from I-kappa B, I showed that in a slide. Now NF-kappa B is free to get into the nucleus. And when it does that, it binds to promoter regions of certain genes that are classified as genes that encode for pro-inflammatory cytokines and chemokines. All right? That's an important take-home message. Okay, what else did we talk about? Yes?
Different components of saliva and your
Right. Right, so I won't go into that, but yeah, and I'm going to continue with it to finish it. So saliva is a great example of a body fluid that has a myriad of different innate response elements. And some of them are redundant in that they will do things that others will do at the same time. So that if there is some kind of strategy by a bacterium to neutralize one of them, the other one is there to protect us. So give me an example of some of the things we talked about.
Like lactoferrin sequesters iron away so that bacteria
Right.
can't use it.
So lactoferrin is a molecule that has the ability to sequester iron, the Fe+2, the ferrous ion. And when it does that, it takes away iron from the bacteria. Bacteria use iron in order to thrive, in order to generate virulence factors. So if you can sequester the iron away from the bug, you've got a great chance of neutralizing that bug. Not completely, but to a great degree. It's far less virulent in the absence of the iron. What's another one? Yes,
Lysozyme.
lysozyme. Now, what does lysozyme do?
It breaks the peptide molecule.
Exactly. At the beta-1,4 linkages between the NAM and the NAG, N-acetyluramic acid and acetylglucosamine, these chains that make up peptidoglycan. So if you can break up that cytoskeleton of the bug, it will implode and die. Back to antibiotics. So there are antibiotics that do the same sort of thing, that have the ability to break that bond. And that bonding process is called transglycosylation. Okay? So there are, did I say antibodies? Antibiotics that break that bond as well. Okay, so there's lysozyme that can do it, and there are antibiotics that can do it. And recall that antibiotics are really bacteriocins produced by bacteria to kill off other bacteria. What else? Yes?
Mucin,
Mucin.
mucin.
Yes. Okay, so this is non-specific. It plays multiple roles, but in the context of innate immunity, it plays two very, very important roles. It adheres to bacteria in fluid phase to do what? Aggregate, and then what? Swallow. So that the net load of bacteria in our mouths is reduced. And only specific, to a great degree, specific bacteria that have a strong affinity for components in pellicle that coat the tooth and, to some degree, the mucoidal lining will bind to. Right? So mucin can bind to bacteria in fluid phase, and mucin can be a member or a component of the salivary pellicle that contains agents that I'll talk about that are anchoring molecules for those early colonizers, those early bacterial colonizers that are deemed beneficial to us. Never in your clinical years should you ever advise a patient to use any of these mouthwashes that claim to promote sterility in the mouth. You know, a patient doesn't know any better and thinks, oh, bacteria are bad. Well, in this case, certain bacteria are very, very good. And the concept that Procter & Gamble and some other manufacturers are putting in the minds of people that you've got to use these mouthwashes in order to get rid of bacteria is a bad thing. Because imagine if you sterilize the mouth. What do you think would happen?
Bad bacteria can take over.
I'm sorry?
Bad bacteria.
Yeah, from the outside. You'd have Staph aureus that shouldn't be in your mouth. You'd have E. coli that shouldn't be in your mouth. These you see in what conditions? Occasionally. What were you going to say?
Oh, I had a question.
Well, wait, hold on to your question. What condition will you see situations that I just described? Yes, exactly. In an immune-compromised condition, all of a sudden, things change. There's instability in the oral cavity, and you'll start to see things like Candida growing. 50-some% of humanity are carriers to Candida, but they're in the yeast form. They're not in the hyphae form. So when they're in the yeast form, they're commensals, they're not really doing anything. The other bacteria are taking over. But in an immunocompromised condition, things shift. And all of a sudden, these yeast can become hyphae, and there are classical ways by which you can see it in the oral cavity. Yes, what was your question?
Oh, what types of mouthwashes do that? Is Listerine?
Listerine, they claim they'll do it. These claims are the things that change people's minds. You know, you're talking about a patient who doesn't know any better, right? Now, patients have become a lot more savvy because they've got the internet, right? But they're coming to you with preconceived notions. And you've got to be able to explain to them that you've got to have good bacteria. Just like you have to have good bacteria in the gut, you have to have good bacteria in the oral cavity. And there are even people who are studying probiotics for the oral cavity, which is something relatively new, the way they're already developing it in the gut. Okay, so I just wanted to, I like to do this kind of thing after a lecture, just to refresh your memory, so that you're with me. Did we talk about histatins?
Yes.
We did? Okay, what did we say about histatins? It's already there.
In terms of the immunoaffinity absorption.
Oh, yeah, okay. So, classic experiment done at BU many years ago by Frank Oppenheim's group. And what they did to identify the fact that these histatins in the saliva were antifungal. They're probably the most important agents, innate response elements, that are antifungal. And what they did is they did immunoadsorption through columns, right? So they took meat saliva, they pooled it for many patients, they passed it through a column that had antibodies to these histatins. And then they let it incubate in those columns. Then they opened the spigot at the bottom, and they let out the saliva that went through that process. And they tested the saliva before they put it in the column and after they took it out of the column. And lo and behold, the saliva that went through the column with antibodies that bound histatin had less than 2% activity. So 98% activity was lost through the immunoadsorption of the neat saliva. So that's pretty good evidence to implicate histatins as antifungal agents. And now there's something called histatin 5, which biotech companies are using to attack both oropharyngeal candidiasis, especially in those cases where you've got denture stomatitis, as well as implant fungal infections. Implants meaning not in the oral cavity, but in the hip. I don't know if I showed you this, but this is an example of the kind of, well, it's in the mouse. And Dr. Pendian, for example, does research in the area of oropharyngeal candidiasis. And she plays with regulatory T cells, tries to amplify regulatory T cells to reduce the inflammatory response due to OPC. But, and she'll teach you more about that when the time comes. This is an example of a mouse model that she works with in oral candidiasis. This is the tongue, a normal mouse tongue. Now, I don't know, can you see these things in purple here? That is infiltration of hyphae of Candida attacking this region of the tongue. And what's interesting is that, and this is a take-home message, is that this seems to be self-limiting in the oral cavity. When you've got candidal infiltration into the tissue, what's going to happen is it's not going to be systemic in the end. It's not life-threatening. It is a quality of life issue. People complain, it burns, it bleeds a little bit, but it isn't life-threatening. And what happens is there is a infiltration of PMNs, like a belt of PMNs that comes in response to an attempt by the hyphae to penetrate further. And that keeps it from getting deeper. Also, if you take the oral candidal species or strains, and you put it in a mouse, more than likely it's not going to kill the mouse. You take candidal species taken from the kidney, systemically infected individuals with Candida, those kinds of fungi can be deadly to those animals. So there's a difference in virulence capability by the oral Candida versus the systemic types. Okay, this is something that's real important. Proline-rich peptides. Proline-rich, so you've got an amino acid in these elements, in these agents are rich in proline. And what happens is a significant fraction of the acquired pellicle is made up of proline-rich peptides. It's important in dictating microbial attachment. That is a take-home message, okay? A very important take-home message. Because proline-rich peptides play a central role in promoting selective adherence of key early organisms, early species of organisms, to the salivary pellicle. Okay? Very important. Proline-rich peptides. Now, this is also really important. There's pleomorphism in this kind of molecule. Now, what does that mean? What do you think it means? When you hear the word pleomorphism?
It can take on different forms.
Different forms, right. And what it alludes to is the fact that biochemists have determined that when proline-rich peptides are in liquid form, they're not recognized well by the adhering bacteria. When they land on the tooth, they have this inherent ability to open up and reveal binding sites for those bacteria. Does that make sense? That's really neat. Yes?
So the difference between pleomorphism and polymorphism, is that poly is genetic and this is just conformational?
In the context of opening up for that kind of interaction, that's pleo. In the context of different structures a molecule can have, that's more like poly.
This is like diagonal.
Well, you're talking about single nucleotide polymorphisms, SNPs,
Okay.
but that's genetic. That has to do with different letters, if you will, in the gene at a certain place that can either encode for something, be nonsense, not do anything, or in the promoter region of the gene, it can be really important in terms of the amount of promotion, the level of expression, right, of the gene. So that's a functional polymorphism. Pleo is, in this case, for a protein to open up, to change its structure so that an outcome occurs, that's pleo. So in the case of PRPs, this thing lands, opens up, is recognized by specific organisms that have a huge affinity, a tremendous affinity for those open epitopes that are normally closed when the molecule is swimming in saliva. So the efficiency of binding is much greater when that molecule lands on the tube. Okay? Now, if you think about it, there's an organism called Streptococcus sanguinis. And you're going to learn a lot about this bug because it is one of the really good bugs. One of the early colonizers, the moment the teeth erupt, not before the teeth erupt. Because before the teeth erupt, different types of streps, like strep mitis and strep salivarius, are the ones that bind to the baby's soft tissue. But the moment the teeth erupt, these guys, proline-rich peptides, play this role, opening up, and now mother-to-child transmission, vertical transmission, happens, and the streptococcus, sanguinis, Actinomyces species, Actinomyces viscosus. These are early colonizers that use the PRPs for binding to the tube. Okay? They have 1,000-fold higher affinity for these PRPs and for the next molecule that I'll talk about, staphyrin, than a bad guy that we'll talk about maybe by the end of today, and that is strep mutans. You've heard of strep mutans? All right, so strep mutans is a streptococcal organism, but its affinity for the PRPs and staphyrin is very, very low, and that's a good thing. The bad thing is, if you put it in the context of an environment rich in sucrose, which we'll talk about, certain virulence factors and certain strategies that it uses that we'll talk about overcome this low affinity for the PRPs and the staph. Am I making sense? Okay. So that's the take-home message. The other thing is, this guy Jeff Gordon at WashU, his claim to fame is studying gut microbes and the importance of the progression of maturation of the normal microflora in the gut with the health of the organ of the host. And he's done a lot of work in mouse models demonstrating the importance of certain early colonizers in promoting the maturation of that epithelium in the gut, so that things like folic acid can transfer across the membrane well for the viability of the host. And what he found was this organ is a Bacteroides thetanomicron. Really neat organism. And this is where there's somewhat similarities between what goes on in the oral cavity and what goes on in the gut. So if you take mice that are born in a bubble, right? Mice that don't see bacteria from the day they're born. Sterile mice. And you give them sterile chow. They won't develop. You look at them, they look macerated, their fur isn't fluffy. The moment he put in this organism, Bacteroides thetanomicron, something happened. And what happened was that the epithelium started to generate certain receptors, proline-rich peptides, on the surface of that epithelium that was used by other organisms to come in and bind. So that's telling you that there's a common denominator here. The delivery may be different, but at the end of the day, the outcome is similar in the sense that you're getting the microflora, the normal microflora, evolving with the expression of these PRPs. Am I making sense? Okay, that's what that's about. Okay, staphyrin. You don't need to know anything about the chromosomes or anything like that. That's not what's important. But it's another molecule that's involved in microbial attachment. That's the most important thing. So the two most important proteins in innate immunity that promote the colonization of good bacteria to the tooth are staphrins and proline-rich peptides. All right, this is taken from Journal Bacteriology back in the late 90s. Paul Kohlenbrander was a pioneer at the NIH, at the NIDCR, who studied the progression of biofilm. We know it in the mouth as plaque, right? And so what he attempted to do was to assess to what degree there was this natural progression of what are referred to as the early colonizers with the late colonizers. Now notice, I don't know if you can see it, but maybe you can see it on your monitor better. These organisms tend to be gram-positives. And these organisms, the late con, tend to be gram-negative. All right? Now, this is where the notion of the anchoring molecules come into play. Staphyrin and proline-rich peptides that I talked to you about, and some other things, but primarily these, are being recognized early on by a lot of these early colonizers. And these are good guys. And these are the guys that keep the exogenous organisms from being able to colonize the oral cavity, primarily the teeth. And these continue primarily as gram-positive organisms. Look at this guy, Fusobacterium nucleatum. Now, you're going to learn more about it in the course with the microbiologist, I think is in the third part of the course, who's hopefully going to focus on this organism because this is a bridging organism. Notice, it separates these gram-positives, early colonizers, from the late colonizers. So it's a bridging type of organism. And Paul Kohlenbrander put a lot of emphasis in understanding the interactions between these guys and these guys through this guy, because this guy is a very sticky bug. This guy has a lot of receptors and adhesins on it that interact with other bacteria. What's referred to as co-aggregation. Co-aggregation. And so there's a lot of interactions occurring. This is just an example of one, and another, and a third, and a fourth, of specific kinds of interactions where specific receptors interact with glycoproteins or sugars, straight sugars, on the other bug in order to cause this coaggregation to occur. Did a lot of in vitro studies to understand the interactions. They used certain enzymes that they identified as destroying, say, the sugars on the surface of one of the bugs so that this interaction no longer occurred. They identified those sugars. And companies like PG and Colgate and others are trying to put these sorts of bits of information into their toothpastes in order to prevent these coaggregations from occurring. It's not all that successful. There's a lot more to it than just that. But this guy does play a very central role in the process that we know as gingivitis. When we go from health to gingivitis to periodontitis, what are we seeing? That in the gingivitis, you still have a predominance of gram-positives in the gingivitis. But if you don't stop it at gingivitis, right, you're going to get periodontitis. Now, in periodontitis, this predominates. And so a very important take-home message is that when it goes from health to gingivitis, you're going from a predominantly 90+% gram-positive, aerobic, fermentative kinds of organisms to less than 90%. Now gram-negatives are starting to come in, but it's still predominantly gram-positive. And if it shifts from gingivitis to periodontitis, and it becomes severe periodontitis, those gram-positives plummet, and gram-negatives go up really, really high. And those gram-negatives start to become, or are, anaerobic and chemoorganotropic. That's a very fancy word for having an organism go from being fermentative, meaning fermenting sugars for their energy source, to breaking down amino acids and small peptides for energy sources. So they are inherently high in what kind of molecules that break down these amino acids? What would you expect these molecules to be? Have you heard of enzymes? Okay, so they have a huge armamentarium of enzymes, unique gram-negatives. So the take-home message, health to gingivitis, still gram-positive, primarily gram-positive, aerobic, fermentative type of organisms. Gingivitis and severe gingivitis, now it's shifting from gram-positive to gram-negative. Periodontitis shifts even more, and now you have a predominance of gram-negative anaerobic chemoorganotropic organisms. So that those organisms are going to destroy the periodontium. They're going to destroy the collagen fibers that interact between the root of the tooth and the bone, right? You're going to get inflammation. You're going to get, a lot of these gram-negatives are going to produce those PAMPs that we talked about that are identified by the PRRs that are going to release NF-kappa B into the nucleus that are going to produce the cytokines. I'm building a story here. Understood? Okay. So that's why this is important. And I'm hopeful that the microbiologist, whom I don't know, is going to be focusing a lot of attention in this dynamic so that you get a better feel for it. By the way, the gram-negative anaerobic kind of organisms, periodontists usually use metronidazole in the severe cases in order to eradicate these bacteria. So these organisms are very sensitive to something called flagell. Another name for it is metronidazole. This is a very interesting slide, and I bring this to your attention. It's historic. It was done in the early 70s at UCLA, where the researchers there asked dental students to wear an EPON strip on the outer aspect of their molars and not to brush for about, I don't know, 4, 5, 6, 7 days. I can't remember the exact number. But why is that important? Because after those 7 days, they took the EPON strips out and they stained what was growing on the surface. Now remember, this is in the 70s. Okay? We know a lot more about what we're talking about now than they did then. So then it was really revolutionary what they discovered. First of all, notice the palisading kind of staining that you have here. Dark, light, dark, light, dark, light, dark, light, dark, light. That's interesting, first off. Anyone venture an idea as to why that is? No, they're all, most of this right here is gram-positive. Okay? Notice, you brought this up. What do you see here? What do you see at this part of the plaque? The tooth is over here, right? Okay, what do you see here? These are different shaped organisms than these. All right? Now this was revolutionary back in the early 70s to discover this. What are these organisms? What is that organism? What are these organisms? That's what I'm showing you in real life. There is a strategy by which this biofilm forms in the oral cavity. This is that bridging organism. These kids were starting to develop gingivitis. All right? And this is predominantly gram-positive. The palisading is due to the fact that these organisms are developing extracellular glucans. We'll talk about extracellular glucans when we talk about strep mutans, because it's an incredibly important virulence factor that it uses to stick to the site it wants to adhere to. Remember, strep mutans, I told you before, does not bind readily to the proline-rich peptides or to the staphnins. So it needs to have a strategy by which it can bind and stay and collect other strep mutans. So the dark ones are water-insoluble glucans that are being formed around these teeth. And the lighter areas are made up of water-soluble glucans, which we'll talk about, which are easier to get rid of. All right, so this is kind of like an introduction to our next topic. Just keep this in mind when we talk about strep mutans and those other things. And this is really something new back in the early 70s when they saw these kinds of organisms appearing at the early stages of gingivitis. Cystatins. What do you need to know about cystatins? Again, busy slide. The important thing about cystatins, they inhibit cysteine proteases. There's a thing called gingipain in Porphyromonas gingivalis. And I'll allude to it when we talk about periodontitis. And gingipain is an extremely important enzyme that's being studied a lot with regard to this organism. It plays a major role in multiple different activities related to dysbiosis. When I started lecturing to you last week, the first slide had 2 reviews, Hashishingalis's reviews, and I urge you to read them. I'm going to go over them, some of the findings from those papers when we talk about perio, but I really need you to look carefully at those papers because it's become a very important topic over the last 5 years. We've now identified organisms in the oral cavity that are very similar to types of organisms that are being discovered in the gut. And these are called keystone pathogens. These are organisms that are commensal-like, and only under certain circumstances, when they increase in numbers, do they have an effect on the bacterial community around them. There's a microbial shift in those organisms that makes them more virulent. The keystone pathogen is doing things like subverting the host immune response so that the other organisms, what are referred to as pathobionts, I'll go over that when we talk about perio, those pathobionts now take over, and they make the situation worse. But the keystone pathogen is the guy that starts the whole process, that shifts the microbial community into a pathobiont community from a commensal community. And it does it by subverting the immune response in the host locally. And what do I mean by subverting the immune response? So you know about neutrophils and PMNs, the first line of defense. Well, it has the ability to inhibit the PMN's potential for phagocytosis. It has the ability to subvert the epithelial cell from wound healing. It has the ability to inhibit the epithelial cell from releasing and producing chemokines to bring in those PMNs. That's called dysbiosis. That's called subversion. And that's why this gingipain plays a role in that, because it can chew up chemokines. And remember what chemokines do. They bring in the immune cells. So here's an organism. One is stealth-like, PG. It doesn't like to be recognized by the host. And it likes to subvert the immune response of the host. So what cystatins do is they attack gingipain, as one example. Anything that is cysteine proteinase produced by organisms is attacked by cystatins. It's not important, all of this stuff, this is for another class. Yes, sir?
Can any of these factors be transferred by HCT in, say, a plasmid or something like this? Or if you have it, then you can avoid
Well,
infection.
remember-
If you don't, then you can't.
Okay, so if you're going to put it in a plasmid, first of all, these are host-related factors. These aren't bacterial-related factors. But, I mean, potentially, if you have the gene, you could generate it in an organism.
We just don't usually.
But you normally will use a bacterial factor, but that's not always the case. You can do plasmids in there, but what would be the purpose of this?
Well, what you'd be trying to do is, in this case, you're trying to avoid detection, right?
You mean the bug?
Yeah, speaking of the bugs, not
Okay.
of
Of the host.
the hosts.
Yeah.
So potentially you could like, I'm just asking, do we see passing on virulence factors as a general rule among the bacteria?
Yes,
or in the mouth?
absolutely.
Okay,
Okay, now you're talking about something really neat. Absolutely. Well, best example is antibiotic resistance.
so that kind of thing, but
Oh, yeah.
to be this. Okay.
Oh, yeah. There are virulence factors. A great example is AA. I don't know if you've heard of it, Agrobacter actinomycetum comitans. It is the agent that is strongly implicated in juvenile periodontitis. And you'll learn about that when you get into pedo. And there is a periodontitis that is seen. There's studies that indicate that the African Americans tend to be more susceptible to it as a race. I don't know if that's true. People from the University of Pennsylvania have done a lot of interesting work in that arena. When I was in practice in Seattle, the University of Washington, I saw a number of families that had a genetic connection to the disease. Some of the kids in multiple families had it. But the thing is, AA, this organism, seems to be playing a very, very important role in juvenile periodontitis. And when I talk about it, it's from ages 4 up to about 15 to 20 years of age. There's an example of an organism that had a bacteriophage, which was a virus, identified as the agent that injected DNA into the AA, serotype B, that made that AA more virulent than before. And that AA produces something called leukotoxin. And leukotoxin is something that kills leukocytes. And so that particular AA is found in kids with juvenile periodontitis. And it tells you how important those leukocytes are in the early stages of defense. So there's an example of a naturally occurring increase in virulence by a virus that attacked a particular species of oral bacteria. And the best example are the antibiotic resistance story that's occurring. I don't have time to talk about this, but quick. There's a lot of studies going on showing that children or babies in the first 3 years of age are, what clinicians are seeing is an increase in gram-positive resistance to antibiotics. And the parents will swear to the fact that they never gave the children those antibiotics. Nor were they kids who had recurrent ear infections. You know the story about babies who have these ear infections, and so a lot of ENT docs, pediatric ENT docs, sometimes prescribe antibiotics. Well, you would expect to have resistant organisms in those babies that have a history of taking antibiotics. It turns out that they're getting it either from their peers in daycare or they're getting it from food. So the poultry industry fattens up its chickens with tetracycline. All right, that's why there's this craze for organic food. No tainting of antibiotics. Long story short, a good percentage of kids now, ages 1 to 5, have a high incidence of antibiotic-resistant gene-expressing bacteria, primarily gram-positive bacteria, in their oral cavities. Exogenously produced, meaning it came in through other sources. And so there is the possibility of bacteria transferring plasmids from one organism to another. But that is a real important point. All right, you want to take a break? and we'll move on to the epithelial-based innate response. All right, 5 minutes? And I've talked about the liquid phase, which is this thing called part of the oral mucosal strategy. I made this up, so whether it's worth anything or not, you tell me. It's somehow the dynamic in the oral cavity may be understood better this way. So there's a fluid phase. I call it that fluid phase, which is the saliva and all the components that we talked about that are in there. I'll talk about secretory IgA as it relates to caries. Then there's a thing called the static phase, which is really that epithelial lining, that barrier between them and us, I like to say. When I was a dental student, a lot of this stuff wasn't known. And this stuff was seen as a simple physical barrier. But we didn't know how it was that when you bit your tongue or burned your palate or bit your cheek, any of that sort of stuff, even aphthous ulcers, how is it that those sites didn't promote massive infiltration of bacteria that promoted bacteremias in us? How is it that happened? So this thing plays a major role in subverting the invasion, if you will, of bacteria when you have wounds, self-inflicted wounds. Even when you talk or when you eat, there are breaks, tiny breaks in this barrier. And we've got to be able to defend ourselves against those. So that's the static phase defense, I call it. EAPs stand for epithelial-derived antimicrobial peptides. And then there's the recruitable phase, which is in the blood and in the vessels, in the periodontium and in the gingiva. Turns out that these guys here that are being produced in the mucosa are playing a role in bringing specific cells from the bloodstream into an area that's needed for defense. There are also resident cells in the mucosa, those dendritic cells that play a role in binding to foreign elements, antigens from bacteria, and they take them to the lymph nodes to activate the adaptive immune system. Those are Langerhans cells. So every now and again you'll see a Langerhans cell in that mucosa. So those are cells that are referred to as antigen-presenting cells, and those are bridging cells between innate and adaptive immunity. You'll also see an occasional lymphocyte, and you'll see PMNs. And I think I mentioned this in the first presentation. This mucosa is unique in that there is a small yet important gradient of IL-8, interleukin-8, in this mucosa. Remember what IL-8 does? What does IL-8 do? Interleukin-8. Why didn't I mention interleukin-8? Yes. IL-8 brings in neutrophils. So what you have here is an active mucosa under normal conditions that has a gradient of IL-8, a normal level of IL-8 that brings in these percolating PMNs into a normal mucosa. That's unique to the oral mucosa because other mucosa of the body don't normally have PMNs unless there's an infection. So that is what I refer to as the static phase. But when that gets compromised in any way, there's a wound, then these EAPs get produced or released because many of the cells in that mucosa have preformed antimicrobial peptides in them that get released upon need. And we'll talk about that next. So why is the mouth so healthy in spite of constant trauma occurring in a very septic environment? And this guy, Michael Zaslav, who was a mentor to me a number of years ago in the 90s, is a pediatrician. He's a pediatric specialist who did a lot of work at the NIH, was always interested in wondering why is it that the skin, he didn't look at the oral cavity, but he looked at the skin. And he used the frog as a great example of being resilient to wounds. So at the NIH, he took frogs, he intentionally wounded their skin and threw them back into this murky environment that had a lot of bacteria and fungi and viruses. And he took them out intermittently and saw that the wounds were healing. But he also did histological analyses and subsequently biochemical analyses and discovered that in the area of the wounds were small peptides being produced by the surrounding epithelium. And when they isolated these molecules, they found that they were antibacterial. They found that they were antiviral in cases where the virus was encapsulated, and they found that it also had antifungal activity. So this is back in the 90s, and it created tremendous interest in these small peptides being produced by a host of different animals. It started initially in insects, and then reptiles, arthropods, mammals, high-order mammals, humans. And we inherited, to a great degree, this kind of activity. So you translate what he saw in the frog, and we went ahead and looked at it in the tongue and looked at it in the oral mucosa, and indeed, these peptides are there. And the peptides react the same way as he saw in the skin of the frog, meaning you wound the skin or you wound the mucosa, these peptides are going to be produced around the area of the wound. Some of them also play a role in wound healing. I may talk about that in regards to the defensin molecules. But he played a very, very important role. And he called this molecule maganin, from the Hebrew word meaning shield, magen. And the rest is history. He created a company in Pennsylvania, Maganin Pharmaceuticals, that you can read about. And so that's where host defense peptides are today emerging from, even though there were entomologists from Denmark and Germany who discovered these things many, many years earlier. But he gave it a lot more oomph. He gave it more applicability, and clearly he instilled this interest in many of us to see what role these things play in human body sites. All right, so AMPs in humans. There are over 45 antibacterial proteins found in human saliva and gingival curricular fluid. To a great degree, these AMPs in human saliva are coming from the epithelium. And I don't have time, obviously, to talk about all 45 of them, or even more than them. The ones that are most studied are the ones in this group. And I don't have time to talk about any of these other ones. I will mention that secretory leukocyte protease inhibitors, or SLIPI, is found in granules of PMNs. It's also found in epithelial cells. And it's got its claim to fame because Wall and Sugars were two investigators who identified their SLIPI's ability to be antiretroviral, which means it could neutralize HIV. The ones that are really, really important, the ones that have gotten more notoriety, if you will, are these. LL37 and human alpha and beta defensins. And I'll talk about these primarily, LL37 and beta defensins. So LL37 is named after the two first amino acids, leucine, and then it's a 37 amino acid peptide. But it comes from a bigger molecule called CAP18, cationic antimicrobial peptide 18. 18 is the big molecular weight of that molecule. And there is a protease that cleaves it and makes it active. The active antimicrobial peptide is referred to as LO37. So that's why that's called LO37. And it's produced, and it's called cathelicidin. Now, it is the only cathelicidin that is produced in humans. These cathelicidin molecules are alpha helical. They're found in pigs, they're found in cows, and those animals have multiple different cathelicidins. We have only one, and it's this one. It's produced in the granules of PMNs, it's found in lymphocytes, it's found in macrophages, and it's found in epithelial cells. And we'll talk about this in regards to certain predispositions to periodontal disease. Then there's the alpha and beta defensins. The alpha defensins, as well as the beta defensins, are beta-sheeted. I'll show you what a beta-sheeted molecule looks like. The human alpha defensins, in difference to the beta defensins, are found in PMNs. So they're not produced by epithelial cells. The beta defensins are produced by epithelial cells. The alpha defensins are also produced by something called Paneth cells in the crypts of lubricones in the small intestine. And they play a major role in eradicating excess bacteria in the gut. If you neutralize the organism, the host, you knock out these molecules from the Paneth cells, you'll get overgrowth of bacteria in the gut. So they play a very, very important role. The beta defensins are produced in epithelial cells, and I'll talk about them in regards to multiple things related to the oral cavity. So cathelicidins, as I said to you, CAP18 is the mother molecule, while L37 is the active part of CAP18. Once it gets cleaved off of the CAP18, it's found in lysosomes of macrophages and PMNs. It's also found in epithelial cells. It can be activated and released from epithelial cells by bacteria, fungi, and viruses, and by activated vitamin D3. Have you had any lectures on vitamin D3? Anything on vitamin D?
A little bit in pathology, blood calcification.
Where?
Maybe it's a little bit, wasn't it? Foundation of calcification.
Oh, okay. Okay, so there's vitamin D3 is really not a vitamin, it's more of a hormone. And you know that it can get activated when you're out in the sun. And, but it gets produced, it goes to the liver and then the kidneys to get the active form, I-125 vitamin D3. I'll talk a little bit about this because it's a really neat way of activating LO37. So I think I have is there a following slide that has a figure on it? The next slide?
Yeah.
No, there's got to be, no, go back. No, that's all? Oh, I added it a couple days ago. All right, I'll show you that slide. I'll update it, I'll send it to Heather and you'll have it. So we'll talk about I-125. So in order to get LO37 to get activated, this thing called proteinase 3 is generated to cleave CAP18 and form LO37. Without this, CAP18 is not active. Now there are people who have shown that CAP18, which is highly cationic, can take LPS, lipopolysaccharide, and bind to it and inactivate it from being pro-inflammatory. That's about all it can do as CAP18. But L37 is antimicrobial. It can kill organisms. It's also activated through the NF-kappa B system that I mentioned to you before. But it's also activated through EGFR, epidermal growth factor receptor. Okay, EGFR is an extremely important receptor on epithelial cells for proliferation. So it plays a normal role in wound healing. All right? So LL37 can get activated through the activation process of epidermal growth factor receptor pathway. So it plays a role in pro-inflammation, it plays a role in wound healing. And it has an important role, most recently, in cross-talking with the adaptive immune system. Meaning, it can play the role as a chemokine. So L37 and some of the beta defensins that we'll talk about play this really important role of cross-talking with adaptive immune system. This is what I meant. So I don't think you have this slide. And why is this important? It's a little busy, but I want you to understand that there's an interplay between the toll-like receptors and vitamin D3 in terms of activating LO37 and the role it plays in further activating, as I said, LO37. And in this case, the example is Mycobacterium tuberculosis, which is going into the cell and playing and liking to live within the macrophage. And tuberculosis, as you know, is the causative agent for tuberculosis. And it likes to live intracellularly within the macrophage. And the macrophage can't seem to kill it. So they discovered there's another way to do that. And this is just an example of what LO37 can do. But the first thing is we know about TLRs, right? We know about toll-like receptors, and we know about PAMPs. So one of the things that I didn't tell you is that when this gets activated, say in a macrophage, vitamin D receptor goes up. Vitamin D receptor. Now, the inactive form of vitamin D, 25D, is in the circulation. All right? And it diffuses through the cell, in this case, gets activated in the mitochondria, becomes I-125 vitamin D, and it binds to the vitamin D receptor. That's what this is about. And it goes in. Now, the vitamin D receptor, in conjunction with I-125, identifies vitamin D regions in the promoter that it binds to activate the gene that induces LL37, or that induces CAP18. This, when I say cathelicidin, in this case, I mean CAP18. Because it then undergoes proteinase 3 cleavage, and LL37 appears, and it can begin to kill Mycobacterium tuberculosis. This is taken from the tuberculosis example. But there's a lot that can be learned from this in regards to the epithelial cell. I couldn't find something that had an epithelial cell story to it. But this aspect of it is common in the epithelial cell. Okay? So what this is telling you is the inactive form goes into the cell, it becomes active in the cell, the I-125 interacts with the vitamin D receptor, and that collectively goes into the nucleus and identifies those parts of the promoter regions of genes that are identified by the vitamin D receptor, VDREs, I think they're called. And cathelicidin gene is one of them that gets activated, and that's how you get eventual LO37 expressed through activated vitamin D. This is an example of what happens in two certain deficiencies that lead to, or two certain syndromes, Morbus-Kosman syndrome and Papilloma-Faber syndrome. I'll talk about them when we talk about periodontal disease, because these two syndromes, common denominators for these two syndromes is the fact that you get really severe periodontal disease. And these happen early on in life. So in Morbus-Kosman syndrome, there's a deficiency of LL37. And in papillon-lefevre, which we'll talk about, there's also deficiency of L37. What this is showing you, have you heard of Western blots? Has anyone done a Western blot? Okay, so what this is supposed to do is you run a gel with your protein of interest, and then you transfer it onto a membrane. And then you come in with antibodies that are incubated with that membrane in order to identify the protein that was originally run in an electrophoretic gel. All right, so it's going vertically one way and then horizontally onto a membrane. So what you're seeing here are Western analyses of neutrophils and plasma and saliva of normal people as well as individuals with Kostman. As you can see here, Kostman, and you've got related controls. And what this is trying to tell you is that the 4.5 or something between 4 and 5 kilodaltons is the L37. The 16 kDa is what they call cap18. And so notice that there's no reaction or very little reaction both in the plasma and in the neutrophils with people with Kostman. That's what that's telling you. And in saliva, again, you can't find it in this Western. Now, if you look at this is a brother and this is a sister. Radiograph of teeth and photograph of gums in patient with morbus Kostman, upper, and her healthy brother. Notice the severe inflammation of the gums. Notice bone resorption. Notice no inflammation, no bone resorption. So that's a very important take-home message, that L37 deficiency predisposes an individual to periodontal disease. Now there's more to it. When we get to morbus Kostman, I'll mention this, but because morbus Kostman has more aspects to it than just L37 deficiency. The next molecule, or the next set of amps that are really important, are human beta defensives. This is what a beta-sheeted molecule looks like, in difference to an alpha helical molecule. Highly cationic, evolutionarily ancient. As I mentioned to you, these molecules were first discovered by entomologists in the fruit fly. Highly cationic, amphipathic peptides. What that means is these peptides have a hydrophobic domain and a hydrophilic domain. And they interact with bacteria because the net charge of bacterial membranes is negative. The LPS, the lipoprotein, lipoteichoic acid, and the gram-positives, they're all negatively charged. These are highly cationic. So the way they interact with one another, that is the molecule and the membrane of the bacteria, is electrostatically. So it's non-specific. The net charge of normal eukaryotic cells, humans, they're zwitterionic, they're non-ionic. So overall, there shouldn't be an interaction, an electrostatic interaction between these amps that are being released and interacting with bacteria. There shouldn't be that kind of interaction with normal human cells. There are exceptions. I won't go into it, but there are exceptions. There are 4 types. These 3 have been discovered in the oral cavity. This one plays a major role in protecting the sperm in the male urogenital tract. HBD1, or human beta defensin 1, is constitutively expressed. It's in the mucosa, the oral mucosa, but it doesn't seem to go up or down a lot in response to challenges. HBD2, human beta defensin, is inducible, but it's inducible through NF-kappa B. Now what does that tell you if it's inducible through NF-kappa B? Inflammation. So it's going to go up when there is a challenge, a microbial challenge. HBD3, of all of them, is probably the most antibacterial because it's got the highest cationic charge. It's plus 11. These other ones are plus 7, plus 5. So it's very, very, very cationic. And you don't need a lot of it in order to kill bacteria. But it also plays a very, very important role in wound healing. And I'll show you where it's normally expressed under normal conditions. But it is induced through EGFR, epidermal growth factor receptor. That's how we know it's involved in wound healing. But the next step is that it's also a bad guy. And too much of a good thing happens in oral cancer. We'll get to that in a moment. They're antibacterial, antifungal, and antiviral. Mechanism of action, I said to you, they interact electrostatically with anionic molecules on the surface of bacteria, whether they're LPS and gram-negatives, LTA and gram-positives. Also, the phospholipid heads of the membranes are phospholipid heads. They're net negatively charged. Phosphoglycerol is an example of a phospholipid head found in bacteria, net negatively charged. We're made up of phosphatidylcholine. We're not charged in that regard. Now, once this thing happens, this electrostatic interaction, the belief is, there's a lot of work being done to identify how this thing is actually killing bacteria. The belief is that they aggregate and form pores. Other people believe that they create carpets, and that pushes the phospholipid heads to the side. And either way, what you get is a membrane that is destabilized. You destabilize the bacterial membrane, you make it prone to implosion. So that's the sort of thing that happens. And also, these molecules have been shown to crosstalk with adaptive immunity. Some of these guys, especially this one, can chemoattract T cells. This guy chemoattracts myeloid cells, okay, like monocytes. So that's the take home with regard to that. This is, we hit on this as we were looking in a scanning EM. This is a gram-positive rod being hit by HBD3. This is what happens when these molecules bind and form that disruption of the bacterial membrane, and you get explosion or implosion, whatever you want to call it. This is just to describe to you that back in late 99, early 2000, they discovered that these epithelial defensins were chemotactic to immature dendritic cells and T cells. Immature dendritic cells. So you know that those dendritic cells that are coming in from outside, they tend to be immature. They have to go through a maturation process when they come in. And to a great degree, that's important. You don't want them to be mature in the blood, just like you don't want monocytes to be active in the blood. You want them to transfer into active macrophages in the tissue. And so the same sort of thing is happening with these immature dendritic cells. They have to mature. They have to be able to present antigen really, really well. So these guys, were HBD1, HBD2, were found by these people to be chemotactic towards these kinds of cells. Now, this is a very busy slide, and just to summarize all the things that we now know about these small peptides, they play a very, very important role in a lot of these functions. Like I mentioned to you, HBD3 causes the recruitment of monocytes and the maturation of those monocytes and immature dendritic cells when they get into the tissue. Just one example of multiple examples. I don't want to go into this. It's much too complex. Each one of these activities can be multiple hours of discussion. This is something interesting. This is expression of HBD2 in normal oral epithelium. This is from the floor of the mouth of the human. Notice the location of HBD3 in the highly proliferating region of that mucosa. Did you get any kind of pathology, physiology in the mucosa already? Do you know that this is the area where stem cells are? Okay, so the stem cells are here. We go through a process of every 10 to 14 days of cells emerging from here as highly proliferating, and then as they ascend, they lose their capacity to proliferate and start differentiating. That's a good thing, because in neoplasia, in cancer, you get a lot of proliferation, with no differentiation. So something goes awry in cells in this compartment. This is called a basal keratinocyte, or stratum basale. So notice that the HBD3 is to a great degree expressed in this highly proliferating region of the mucosa. And as the cell ascends, it starts to express HBD2. So you're not seeing HBD2 expression here, you're starting to see it in differentiating cells. And as it ascends even further, it undergoes apoptosis, and then it exfoliates. Remember the stratum corneum, there's a very, very thin layer of stratum corneum here, of dead cells. Eventually it exfoliates. Now there are regions where that stratum corneum is thicker and thinner. It's not anywhere near what you see in the skin. But this is an example of what you see under normal conditions. But notice what happens under abnormal conditions. This is an example of a carcinoma in situ. Stage 0 by pathologists. If you can identify this in a patient's mouth and get him, or do it, or excise it yourself, or get him to an oral surgeon in time, you'll save this person from having to go through severe surgeries. Because this is stage 0. You remove this, the person is healed. The moment it becomes stage 1, this capsule bursts, and you're getting cancer cells immersed, metastasizing to different parts of the mucosa and out into the lymph nodes. What I'm trying to get at is notice how much HBD3 there is here, because this whole area is full of proliferating cells with no differentiation. And one way to determine it is there's a target called PCNA for highly proliferating cells. And it's this is immunofluorescence. I forgot to say this. And so these are antibodies that are fluorescing, either pink, reddish, or green. And if you have two things co-localizing, the red and the green PCNA, they're using antibodies with green fluorescent light. So if the red and the green come together, you're seeing an orange, right? Or yellow-orange. And that's what this is representing. This is in this area. So what this is telling you is that this is highly proliferating. Now, how do you know it's cancer? Well, there's something called beta-catenin. It comes from the Wnt system. There's translocation of beta-catenin from the cytoplasm of the cell into the nucleus. That is a signature thing for neoplasia. Notice the nuclei here versus the nuclei here. Notice how clear, translucent the nuclei are. This is DAPI, it's called DAPI. It's a blue kind of stain. But notice the kind of response you get in nuclei here. They're more veil-like. So this is telling us this is neoplasia. This is telling us there's a lot of proliferation occurring. Notice that even in the normal site, this is normal. This is somewhere over here. You expect this to have proliferative capacity, right? So what this is telling you is HBD3 is overexpressed in early stages of cancer. And because it has the capacity to chemo-attract myeloid cells from the bloodstream, you also see a tremendous amount of what's referred to as CD68 positive cells. All of this are CD68 positive cells next to the area of the carcinoma. It's called carcinoma in situ local. It doesn't move. It hasn't opened up. It hasn't metastasized. It's stage 0. So CD68 cells are cells that are expressed on the surface of monocytes and macrophages. So this is telling you that HBD3, which has the capacity to chemo-attract these myeloid cells, are bringing in these guys and in high numbers. Notice CD3, which are T lymphocytes, It's not bringing in T lymphocytes. It's bringing in these myeloid cells. Now, why is that important? Because these myeloid cells are highly highly inflammatory. The other thing that was discovered is not only does the HBD3 bring it in, but it promotes the release of pro-inflammatory cytokines from those CD68 positive cells. So a microenvironment is induced through HBD3 to promote more neoplasia. All of that is a bad thing in the context of cancer. Am I going too fast, or are you understanding this? Come on, tell me, because I can repeat it.
Yeah.
Repeat it? Okay. So, did you get the part under normal conditions where the HBD3 is expressed in the basal keratinocyte region, and how as the cells ascend from proliferating to differentiating, it shifts from HBD3 to HBD2? Did you get that? Okay. Did you get this transition that we were able to discover from normalcy to carcinoma in situ? And you saw a lot of HBD3 in the carcinoma in situ? You saw that. Okay. And you saw the beta-catenin was able to demonstrate neoplastic events in that carcinoma in situ because of the veiling effect, the translocation of beta-catenin from the cytoplasm into the nucleus. And you saw the pcDNA was able to demonstrate proliferation, a lot of proliferation in the carcinoma in situ region. Did you see that? Okay. What this is demonstrating is what is the outcome of overexpression of HBD3 in the context of cancer. Because I'm trying to tell you, there are certain things in innate responses that are good, and in the context of cancer, we're discovering are not so good. So HBD3 overexpression, because it plays a role as a chemokine in carcinoma, and this is squamous cell carcinoma, right? Oral squamous cell carcinoma. And CIS, carcinoma in situ, is the earliest stage of squamous cell carcinoma. So, overexpression of HBD3 in carcinoma in situ is already promoting a microenvironment through the selective chemoattraction of those myeloid cells, which are CD68 positive, into that area, and stimulating them to release chemokines and cytokines, primarily pro-inflammatory cytokines like IL-1, IL-12, which exacerbates the environment and makes the tumor more aggressive in the early stages. Does that make sense? More sense? Okay. Okay, I don't know if I want to go into this. So why is the mouth so healthy in spite of constant trauma? Why are HBDs constitutively expressed in oral mucosa? The answer may lie with some of the ubiquitous bacteria in the mouth that induce epithelial cell-derived antimicrobial peptide. So the take-home with this, without having to go into the slide, you can look at the slides. I don't want to spend more time on this. So there are bacteria, it turns out, that can interact with the epithelium to promote the expression and release of these amps. And then there are bacteria that don't. So an example that I put in these slides is that Fusobacterium nucleatum is an example of a gram-negative bacteria that can promote the release, the expression and release of HBD2 really well. Remember, HBD2 is an antimicrobial peptide released by epithelial cells. On the other hand, Orphomonas gingivalis appears not to be able to do that. And remember, in the context of PG, we're talking about a stealth-like organism, doesn't want to be recognized by the immune system, under the radar screen, and clearly doesn't want to promote the release of amps that will kill it and kill other bacteria. It has a strategy of its own. So in the slides you'll see, okay, from a PCR perspective, HBD2 is induced by FN, HBD2 is not induced by PG. And then when you look at, interestingly enough, what is the sensitivity of HBD2 and HBD3 in terms of their ability to kill these different species of bacteria. It turns out that PG is a lot more sensitive to the killing of these HBDs than FN, which is telling you that there's a strategy that may have evolved evolutionarily by FN to interact with the mucosa to kill off other bacteria in order to maintain itself. Yeah.
Yeah, with these ubiquitous, the normal flora, is it of greater importance that they're inducing these antimicrobial peptides, or is it of greater importance that owing to their pellicle adherence, they're out competing the others? So is it more about these peptides or more about the competition?
It's a dynamic that includes both of those. The gram-positives are doing it through selective affinity towards those anchoring molecules that I mentioned. In gram-negatives, it's a totally different story. And that's why I'm introducing this as a way by which the gram-negatives may have a strategy to promote the release of host response amps to kill off other bacteria so that they can thrive. All right? So that's essentially what this is all about. So I don't want to go into those slides, and we don't have much time, and it's 10 of 12. So all of this stuff is just telling you the FUSO, the porphyromonas, giving you sensitivity levels, what happens when PG comes in contact with recombinant HBD2. And notice FN, on the other hand, has this incredible ability to sequester the molecules away from the membrane so that it can thrive. So those are things that you can see. And then a high magnification shows you exactly that these dots, these things here, are essentially gold particles attached to antibodies. And those antibodies are binding the HBD2. Notice the membrane of the FUSO is here. And you see these amorphous structures coming off of the membrane. And notice they're being sequestered away from the membrane. So there's a strategy here that's really neat that FN has that PG doesn't have. FN can induce the expression of these things, and FN then has a way by which it can protect itself from what it's inducing. Anyway, long story short, that's it for innate immunity. So I'm a little bit behind, but that's okay. We'll talk about caries next time. Do you have any questions? Anything that I can answer while I'm here? We normally end about 10 up, right? Okay, so we did it on time. If you have any questions or anything, contact me. I'll be more than happy to respond. And do yourself a favor. Come to class. All right? Please. Thank you.